Hello, I'll introduce the paper submitted to iMeta. The title is Spatial Multi-Omics Identifies a NOTCH3-Mediated Capillary-mCAF Crosstalk Driving Immune Exclusion in Hepatocellular Carcinoma. Fibrosis is considered an early stage in the development of liver cancer. The bidirectional regulation between tumor cells and fibroblasts is a key factor in further mediating disease progression. Recent studies have shown that tumor-associated fibroblasts are a heterogeneous cell population with diverse functions and spatial distribution. However, there is still a lack of high-quality research data regarding their functional polarization characteristics and spatial distribution patterns in hepatocellular carcinoma. In this study, we built a spatial high-resolution multi-omics atlas of HCC and uncovered the DLL4 NOTCH3 axis driving fibrosis and immune exclusion. Integrating pan-cancer scRNA-seq data, we identified mCAF collagen polarization as a key driver of immunotherapy resistance. Targeted NOTCH3 alleviated fibrosis, enhanced T-cell infiltration, and synergized with immunotherapy, offering a potential rationale for combination therapy in HCC. Single-cell spatial data reveals HCC landscape across tumor, boundary, and normal regions. Using COSMX SMI platform, we precisely matches He, If, and spatial transcriptomics data in the same slide. Spatial data reveal tumor cell expression heterogeneity spatially and a series of prognosis-related genes have been identified. Spatial data reveal tumor cell expression heterogeneity spatially and a series of prognosis-related genes have been identified. These results highlight the prognostic value of CAF functions. Subclustering all CAFs reveals functional and spatial heterogeneity across tumor, boundary, and normal regions. Specifically, matrix-producing mCAFs correlate with poorer outcomes, whereas inflammatory iCAFs correlate with better survival. Spatial transcriptomics and proteomics reveal signature genes for distinct CAF polarizations. Spatial transcriptomics and proteomics reveal signature genes for distinct CAF polarizations. Spatial ligand receptor analysis reveals DLL4 NOTCH3 crosstalk between capillaries and mCAFs, forming spatially adjacent profibrotic niches. H&E staining, spatial FISH, and spatial proteomics analyses confirm that capillary-derived DLL4 triggers mCAF collagen polarization. In vitro knockdown experiments show that NOTCH3 is crucial for driving fibroblast collagen polarization and subsequent immune evasion. In vitro co-culture models also indicate that the DLL4 NOTCH3 axis promotes collagen matrix formation. CD8/CD4/DC immune triads aggregate at the invasive boundary to trigger immune activation, but mCAF enrichment blocks their infiltration, and mCAF macrophage spatial colocalization further drives immune evasion. These results suggest that mCAF deposition in the TME not only forms a physical barrier but also induces immune cell anergy, facilitating tumor immune evasion. Finally, orthotopic mouse models demonstrate that NOTCH pathway inhibition synergizes with immunotherapy to promote immune activation. Furthermore, spontaneous HCC mouse models confirm that targeted NOTCH inhibition sensitizes immunotherapy, offering a potential molecular target for clinical translation. Overall, this spatial multi-omics study reveals how capillary-derived DLL4 triggers mCAF collagen polarization via the NOTCH3 signaling axis in hepatocellular carcinoma. This crosstalk creates a fibrotic barrier, driving immune evasion. Crucially, inhibiting NOTCH3 reverses this polarization, enhancing T-cell infiltration and sensitizing cold tumors to immunotherapy. Thank you.